Good morning, and welcome back to the MCAT grind. Today we have Biochem Chapter 3, Non-Enzymatic Protein and their analysis. So, starting off, we have cellular proteins. That includes the structural ones, including collagen and elastin, which are found in your as connective tissues. They're in an extracellular matrix. Collagen is, you might hear it from helping your stability and structure and strength. So there are also tablets you can eat to help with collagen, but it's also found in fish and like the bone marrows too. Elastin is also in connective tissue, and it's more for recoiling back to the same place. So collagen more for structural strength and flexibility, while elastin is recoiling back to where it needs to be. Keratin is an intermediate filament, and it's present in your hair and your nails and for structures. Actin is part of your microfilaments, and actin and myosin acted together helps with your muscle contraction. So it's in the myofibril. It's in the thin filaments of the myofibrils. Tubulin is part of your microtubules, and tubulin functions to help with the movements, including in the cell division process and to migrate the parts away. And they're like the structural components to the movement. Think about little tubes or cable wires. The motor components, including myosin, which are little heads and necks. They're like little giraffes, and what they do is go whoop, whoop, and they pull the actin, and that's contraction of the myofibril. Myosins are the thick heads of the myofibril. So they're in their microfilaments as well. Kinesin and dynein, they are positive and negative parts of the microtubules function, and they kinesin pulls using the tubulin, pull it towards the positive end, and dynein pull it towards the negative end. So in cells when they're separating in mitosis, kinesin pull the components towards the poles, and dynein pulls the wastes and recycle them towards the center. Binding proteins include like DNA binding proteins, oxygen binding proteins, hemoglobin and stuff. They all help sequester and transport proteins as needed. Without them, you can't have any proteins being moved around within your cell. Cell adhesion molecules, or CAMs, they include cadherin, integrin, and selectin. Cadherin are mostly for cell-cell binding, so they bind cell and cell adjacent ones to each other, like epithelial cells, and keep them binded. Integrins are found on the surface of cells, and what they do is they help with the movement of white blood cells and certain cells, and they're found on the surface, and also help with host defense. Selectins are found on the surface of white blood cells and epithelial cells as well, but it's bound to the a glucose chain or a glycoprotein chain. So it's the weakest bond of adhesion. Antibodies, you always know this, they target antigens and they help with opsonization, which means recruiting white blood cells to come and attack them, as well as inactivating the pathogen. Also, what they do is they agglutinate the pathogen. Biosignaling, we have ungated channels, which just diffuse down a gradient. Voltage gated is once it reaches the depolarization threshold, the gate will open. Ligate gated Once a ligand attaches to the outside, it will open, allowing content to flow in. Enzyme linked, there's a ligand binded to the outside and it triggered a secondary messenger cascade. And that cascade itself includes PIP2 and your IP3 and so forth, and that triggers another enzyme function. G protein coupled receptors uses a G protein, which is combined with the trifecta of beta, alpha, and gamma. The alpha component is normally bound to GDP, and once it's stimulated from the outside, the membrane side, inside the intracellular side, it will have binded to a GTP instead and knock off that GTP, and the alpha component will go ahead and either stimulate, inhibit, or trigger another component of adenylate cyclase, and then once it's done, it comes back to bound with GDP, and then bind back to the beta and alpha, beta and gamma component, and back to native state. Next, we have isolating protein functions, or isolating proteins. We have polyacromide gel electrophoresis. Well, that gel electrophoresis is something like a gelatin or like a gel, and most of you will probably have done this already. Native gel keeps the protein in its native state and let it run through the gel. Problem with that is that many proteins share so many charges so that it'll pretty much equal, and their sizes are also pretty similar, so it's kind of hard to tell. However, the good thing is you can recover the protein after the gel is run. SDS, sodium dodecyl something. SDS, basically, an SDS page, it will neutralize all of the covalent bonds and make them basically all the same charge. So the only thing that's limiting them, or making them different, is their size of the proteins. So SDS basically makes all the charges the same. And then, it's something sulfate. Sodium dodecyl sulfate. Neutralize all the covalent bonds and allow them to separate by their size only. In isoelectric focusing, we have anode to cathode, and this is counterintuitive, but here we have acidic at the anode and basic at a cathode. Acidic, we have positive because more protons. And basically, remember back when we did our Biochem Chapter 1 unit, we have our amino acids. All amino acids have different PIs, or their pH, at which point they're neutral. If we have a compound that has different functional group, that may change their PIs. Now, they will move accordingly to the isoelectric until they move to a PI, and if a amino acid is more basic, they will move more towards the cathode, and so forth. Chromatography, we have size, column, and ion exchange. Size exclusion chromatography, in it, the small ones, the smaller proteins will get stuck in the pores, thus the larger ones will actually go past. So this one's counterintuitive because the smaller the size, the slower it goes. It will get stuck in the gels. And the larger ones will pass through around easily. Column chromatography, you have silica beads, and that silica beads are polar, so the polar ones will get stuck and the non-polar ones will travel down faster. In ion exchange chromatography, you have charged ions, and it will, depending on the stationary phase, it will bind the protein more. So if you have a positive ion in there, it's going to bind to the negative ones, and the positive ones will run down faster. We have ways to determine the protein structure: X-ray and NMR. The X-ray is the one that's used mostly, 75% to determine the proteins, and nuclear magnetic resonance get about 25% of our proteins we find today. To determine the concentration, we have Bradford assay as well as UV. UV just self-explanatory, you shine UV light through, and it's a common machine that's big, and you put in multiple vials that have different, that have a standard. You use a standard and you shine through, get a certain concentration, label it, and you can plot it out into a curve and see what your unknown concentration is. Bradford assay, you will just add a chemical that's brown, and once you add a protein in, you'll get certain concentrations of blue as it binds more and more to the protein. All right, and that's all, folks.